こんにちは。静岡県立大学茶学総合研究センターの雲野恵子と申します。今回は日本緑茶の認知症予防効果についてお話しさせていただきます。
This graph shows dementia patients in Japan grouped by age. Among women, over 50% have dementia in their early 90s, and over 70% have dementia in their late 90s. In men, on the other hand, about 40% have dementia in their early 90s, and over 50% have dementia in their late 90s. You can see from this graph that more and more persons have dementia as they get older. This means that aging is the biggest risk factor in developing dementia. In Japan, being in your 90s or 100 years of age is said to be the 100-year life, meaning that a life shift is important. Even though the risk of dementia rises sharply nowadays, if we could maintain our brain age, for example, to that of a 70 or 80-year-old, then we could drastically lower the risk of dementia, even if our physical age is in the 90s or 100s. But is such a thing really possible? Genetic predisposition, or namely your genes, play a role in dementia, yet, as just mentioned, the major risk factor is brain aging. Stress and a poor lifestyle also act to speed up brain aging. This means that the outbreak of dementia can be delayed by reducing stress and finding ways to fight aging of the brain. Well then, how does drinking green tea actually help prevent dementia? Let's take a look at results gained from disease research studies. These results from Tohoku University show that persons who drank 2 or more cups of green tea a day had a lower risk of dementia than those who didn't drink it. However, these good results weren't found among those who drank black tea, oolong tea, and coffee, etc. A look at Kanazawa University research results from a 5-year follow-up study of about 500 persons aged 60 or older showed that 26 persons had dementia and 64 persons had mild cognitive impairment. On checking who drank green tea and who didn't, results clearly showed that those who drank green tea every day had a lower percentage of dementia or mild cognitive impairment compared to non-green tea drinkers. This shows that drinking green tea daily is effective in preventing dementia. Green tea contains an astringent or slightly bitter taste component called catechin, a bitter component called caffeine, and an umami, which is flavorful component called theanine. So how do these ingredients relate to dementia? At this point, I want to talk to you about 2 of these components, or namely the suppression of brain aging by green tea catechin and the same effect by way of stress reduction of theanine. First, talking about catechins. In an animal experiment, mice drank green tea catechins daily. The catechin concentration was diluted to 3 times weaker than the concentration we drink in our green tea. Young mice had good memories and exhibited high memory retention, but as shown by the black bar, as the mice got older, their memory retention deteriorated. However, as you can see in the green bar, the drop in memory retention was suppressed in mice who drank catechins. This result shows that catechins do in fact exert an effect on the brain. Well then, do catechins actually exert effects on nerve cells of the brain? Carrying catechins to nerve cells that are the actual substance of the brain by way of blood vessels extending throughout the brain requires, of course, that the catechin leave the blood vessels. However, these blood vessels are formed very intricately in a structure that keeps any non-essential substances from penetrating into the brain nerves. This structure is called the blood-brain barrier. We carried out the following experiment to find out how much of the catechin actually gets to the nerve cells of the brain. This model was made to simulate this blood-brain barrier, or simply BBB. By investigating the flow to the brain from the blood vessels over a certain amount of time, we found that catechin does indeed get to the brain. Among catechins in green tea, a catechin called Epigallocatechin, gallate, or EGCG is present in the largest amounts. We found that it takes about 30 minutes for 4% of this EGCG to pass through the blood-brain barrier, or BBB. This 4% is certainly not a large value, But we know that catechin is definitely conveyed to the nerve cells of the brain as EGCG. The green tea we drink is carried as EGCG to the intestines by way of the stomach. About 0.1 to 2% of the EGCG from the amount you drink is taken into the intestines and eventually arrives at the brain by way of the bloodstream. Our estimate from the above experiment with the BBB is that about 1/10 of this EGCG in the blood is then taken into the brain. However, most of the EGCG is decomposed, or namely broken down in the intestines. In the small intestine, the EGCG is broken down into Epigallocatechin, or EGC, and gallic acid, or GA. And in the large intestine, the EGC is further broken down into a substance called dihydroxyphenyl Y-valerolactone. This is called EGCM5, which further becomes glucuronic acid and sulfuric acid conjugate. We confirmed that these substances were definitely able to pass through the BBB. We next investigated how EGCG and substances broken down from it that reach the nerve cells of the brain actually exert effects on those brain nerve cells. Results clearly show that when EGCG is placed in a culture medium, nerve cells, which usually have almost no spikes or protrusions, develop extremely long extending protrusions, which are differentiated nerve cells, or namely nerve cell that attained an active state. We also discovered that EGCM5 provides the same effect. This means we now know that EGCG and its metabolites, or end products, work to activate the nerve cells. To sum this up, catechins such as EGCG taken into the body as green tea first arrive at the small intestine, reach the brain by way of the bloodstream some 2 to 3 hours after the EGCG was taken in, and then activate the nerve cells of the brain. After that, the EGCG dissipates away, and after a further 8 hours have passed, the EGCM5 from the decomposed EGCG is now taken into the large intestine. The EGCM5 carried along the bloodstream then arrives at the brain, and this substance activates or reactivates the nerve cells of the brain. The current view is that both EGCG and its metabolites in this way act to suppress the decline in brain functions that occurs as we become older. The green tea we usually make in a teapot contains about 70 milligrams of catechins per cup, and the green tea from one PET bottle contains about 50 milligrams of catechins. Our view is therefore that drinking several cups of green tea or one PET bottle of green tea every day serves to activate the nerve cells of the brain. Now let's talk about the effects that theanine provides. I set up this experiment because I wanted to clearly show the bad effects that stress has on the brain. Mice are usually raised in groups, making them feel that other mice are their companions. We placed partitions in the cage, dividing it into one compartment for each mouse. After raising them separately in this way, the mice developed awareness of their own territory in the case of male mice, and when the partitions were removed after 1 month, the mice felt stress at finding other mice inside what was their own territory. We then investigated the effects on lifespan and brain aging after raising them in this type of situation. In applying stress by face-to-face encounters in this method, we found that mice that at first glance appeared to normally get along well together tended to get irritable over trivial incidents and start fighting among themselves. We often feel stressed out lately because we cannot go outside often enough due to the coronavirus and have to do more and more remote work. Maybe we are in much the same situation as the test mice. Mice raised under these conditions where stress was applied had an average lifespan of only about 14 months compared to normal mice raised in groups who had an average lifespan of 18 months. The stressed mice therefore died earlier with their lifespan shortened by about 4 months. However, when mice raised under the same conditions of applied stress drank water containing theanine every day, their average lifespan was 18 months. In other words, their lifespan was extended to the same amount of time as the normally raised group of mice I mentioned. These results show that theanine is capable of returning lifespans cut short by stress back to their original state. Investigating brain atrophy to find effects on brain aging revealed that when stress was applied by raising mice face-to-face, this stress did indeed accelerate atrophy or shrinkage of the brain, but the group that drank theanine was able to recover from this stress. In learning ability in mice, some were quite unable to learn as the time grew longer, and the longer the time, the more their learning ability deteriorated. Learning ability was found to deteriorate when mice were subjected to stress, but those mice who drank theanine were found to have improved learning ability. These findings show that brain atrophy and stress accelerate brain aging, but that theanine can suppress those aging effects. These dementia preventive effects of Japanese green tea can be summed up first of all by the effect of suppressing the age-related decline in brain functions through use of Epigallocatechin, gallate, or namely EGCG and its metabolites. Another effect is suppression of accelerated brain aging from stress achieved by theanine, and its dementia preventive effects are shown. The health benefits of green tea catechins were recently described on a show called Medical Frontiers, produced by NHK World Japan. So please check it out if you feel interested in this topic. This completes our description of the dementia preventive effects of Japanese green tea.
日本の緑茶ですとカテキンが溶出しやすいということがありますが、中国茶のようなお茶でも濃度が薄くてもトータルで摂る量が一緒だったらもしかしたら一緒かもしれません。また今回はお話ししなかったのですが、テアニン以外のアミノ酸にアルギニンというのが日本緑茶には高く含まれております。でこのアルギニンはテアニンと同じようにストレスを軽減する作用がありますので、このアルギニンが高い日本緑茶っていうのはちょっと優れている点かもしれません。はい、先ほどの疫学調査の結果から考えますと、たくさん飲まなくても1日数杯飲めば良い効果を示すのではないかと考えられます。ただし毎日飲むということがとても重要なことのようです。本日は動画をご覧いただきましてありがとうございました。